Welcome to Brilliant Minds Unlocked. Today we are taking a deep dive into a world that's, well, often overlooked, a quiet field of maze, to uncover the secrets of one of history's most visionary scientists. Imagine standing alone, in a silent cornfield. For most of us, those tall, swaying stalks are just agriculture. But for Barbara McClintock, that corn was practically whispering profound secrets about life itself. It was an entire universe just waiting to be understood. So our mission today is a deep dive into the life and the truly radical research of Barbara McClintock, the genius geneticist who discovered what we call jumping genes. We're going to unpack this story, but also the immense resilience it takes to pursue an idea when, well, the entire scientific world rejects you for decades.
And that rejection is really the core of her story, isn't it? McClintock's work wasn't just adding a little footnote to biology, it was a direct challenge to the very bedrock of 20th century genetics, the idea that genes were stable, fixed things.
Yeah, so in this deep dive, we're going to help you understand exactly what she saw in that corn, why it was dismissed for so long, and really the powerful lesson her perseverance offers about trusting your gut, even when you're completely alone.
Okay, let's start at the beginning of that journey. She was born in 1902. And from everything we've read, she was just fiercely independent and curious from the start. The sources paint this picture of a mind that, refused to stay silent.
You really have to see her story through the lens of that era. I mean, we're talking about a time when society expected women to fit into these, the source calls them fragile boxes.
Right.
Domesticity over discovery.
Yeah.
She loved science, even when her teachers were telling her it was too difficult for girls. But she just didn't see those barriers, did she? She just saw intellectual puzzles to solve. She goes to Cornell to study botany at a time when very few women were treated as equals in those labs. And here's the paradox. Her brilliance was so obvious, but that brilliance came with resistance. Her drive was seen as strange, even unfeminine.
That's such a key point. Her exceptionalism guaranteed her isolation, but, and this is critical, that isolation became her strength. She wasn't being pushed and pulled by whatever the mainstream idea was at the time, so in the quiet of her lab with her microscopes and her seeds, she could just observe. She developed this patient way of looking at life that, well, it let her see things nobody else was seeing.
So that solitude, that unique perspective,
Right.
Where does it lead her? What exactly was she looking at for all those hours? It was maize, corn. She chose it because its chromosomes are large and relatively easy to observe. So she's looking closer and closer at these spiraled threads of genetic information, and she starts noticing patterns, strange variations.
And this is the moment, the breakthrough. She wasn't just seeing abstract patterns. She was seeing the real, visible results of genetic movement. Think about the corn kernels themselves. If genes were fixed, a kernel should just be one color, right? Yellow, maybe, because the gene is static. But McClintock saw speckled kernels, spotted kernels, striped ones.
Variegation.
Exactly. She realized the gene for that color pigment was being switched on and off, or it was literally jumping out of position, which caused that mottled look.
So the speckles on the corn, that was the movement. That was the proof. A gene was literally moving, creating spots as it switched on and off.
Precisely, and that's why she called them transposable elements.
That is the breakthrough.
It is. She saw genes not as these fixed soldiers in a perfect line, but as nomads, shaping life through motion and change.
Okay, so to really get the scale of the backlash she faced, we have to understand the scientific dogma of the day. Genetics was built on this idea of stability.
Complete stability.
Genes were supposed to be like lines carved into a stone tablet. Predictable. Precise. That was the whole foundation of the field.
And it's so important to stress just how big a challenge this was. By saying genes could move, she wasn't just tweaking a little detail, no. She was basically saying the entire infrastructure of inheritance, how traits are passed down, was dynamic, maybe even chaotic.
It was an intellectual confrontation.
A huge one. And the scientific establishment, well, they reacted exactly how you'd expect.
So she presents her findings in the 1940s, early 50s, and the reaction from the leading geneticists is just disbelief.
Total disbelief.
They called her confused, her work too complex, or just plain wrong. We even know from the records that some major figures in the field literally laughed at her presentations.
And the professional cost of that? It was immense. She was living so modestly, dedicating her entire life to this work purely for the discovery. So to have it met with ridicule, that rejection was deeply personal. It's the kind of thing that would have crushed most scientists.
But this is where her story really becomes something more than just science. Instead of giving up or fighting back, she showed this incredible resilience. It wasn't about professional pride. It was just a profound certainty in what she had seen.
She didn't need their recognition because the joy was in the discovery itself. She saw that the world just wasn't ready to listen. So as journals turned down her papers and her peers kind of questioned her sanity, she pulled back. She withdrew from the noise.
And the sources say she did this not in defeat, but in peace.
Right.
She just went back to her faithful collaborator, the maze.
Mm-hmm.
And it's that purposeful isolation that I find so fascinating, the freedom it must have given her.
That's the key to everything. While everyone else doubted her, she just kept observing, kept refining her models, connecting patterns no one else had the patience to see. She was making some of the most profound insights in genetics because she was outside that mainstream pressure cooker. Her solitude was fertile ground. She was just quietly confirming that life is this dynamic, adaptable, resilient system that's constantly rewriting its own code.
And decades passed. Her work just sat there, largely ignored, until the rest of science finally developed the tools to catch up to her. It wasn't until the 1960s, maybe the 70s, with molecular biology really taking off, that other scientists started finding evidence of these mobile genetic elements. First in bacteria, then viruses.
And slowly, almost reluctantly, her once radical vision started to become a cornerstone of modern genetics.
Which brings us to the ultimate vindication.
Yes, 1983. At 81 years old, she's awarded the Nobel Prize in Physiology or Medicine.
And what's so significant about that is she was the first woman ever to receive it in that category alone, without sharing the honor.
It was a lone recognition for a lone genius. And when she stood on that stage, there was no anger, no bitterness about the lost decades.
Just this quiet, knowing dignity.
Exactly. She always knew she was right. It just took the world a very long time to see what she saw.
And the impact of jumping genes today is just everywhere. It revolutionized our understanding of evolution, genetic diversity, how cells mutate.
I mean, it informs so much of modern cancer research, biotechnology. Any study of the genome today carries the shadow of her insight.
So her story becomes this powerful lesson that really goes beyond science. It's about courage, patience, and just having absolute faith in what you observe.
It reminds you that solitude isn't failure, that silence can actually be the very thing you need for a profound idea to grow before the world is ready for it.
So what does this all mean for you, the person listening, maybe facing skepticism in your own field?
I think the core takeaway is that discovery isn't a straight line. It's not always met with applause. It's about being willing to see movement where everyone else sees stillness.
Yeah.
So when you face that doubt, when other people don't see the variegation in your own work, think of her. She waited decades but never stopped believing in the truth of her evidence.
And that leaves us with a final thought, a kind of challenge for you. What if the world's next great discovery depends entirely on someone who is patient enough and confident enough to refuse to give up? Someone just like you.
Greatness rarely announces itself with a big fanfare. No, it grows quietly, like a seed under the soil just waiting for the right moment. The goal should be a connection with your work, with the truth of it, not the immediate recognition.
Her greatest legacy is that reminder that life itself is never static. It leaps, it adapts, it transforms, just like the genes she discovered. And just like the people who are brave enough to follow their own path to the truth.